All right, good afternoon, everybody. It's a great pleasure and privilege to introduce today's speaker, Simon Boulton. And before I introduce him, it's also my pleasure to acknowledge the opportunity we are given to have Simon here by the Story Endowment, who allows us to invite one speaker for a student seminar, and this is a seminar in chromosome biology. And the organizers of the seminar thought a very long 1 millisecond about the person who we wanted to have here for this special seminar, and that's Simon Boulton. And in an hour from now, you will understand why it took us only 1 millisecond to decide this. Simon is truly a rising star in molecular biology. He has won the gold medal of the European Molecular Biology Organization a few years back, and he continues to accelerate the pace and significance of his research. He obtained his undergraduate education in Scotland at the University of Edinburgh, and then his PhD at Cambridge with Steve Jackson, and he started to work on genome integrity and maintenance of genomic stability ever since. He then moved to Boston to Harvard Medical School to work with Mark Vidal, and sort of got exposed to a completely different set of tools, and picked up the genomic toolbox in Mark's lab and another model organism, moving from Saccharomyces cerevisiae budding yeast to the roundworm Caenorhabditis elegans. And it is really the ease with which Simon moves between systems, yeast, worms, mice, human cells, and the ease with which he's using different approaches from biochemistry, genetics to cytology that typifies his work and makes it so interesting and important. Simon came back to Europe, or went back to Europe, to the Cancer Research UK 10 years ago, and built up his own research program. He has made numerous important contributions, discovered several new genes that are central players in genomic stability, including RTEL, which we will probably hear today about. He just published a beautiful paper, and I can say that because we had it in Journal Club this morning, on the function of MMS19 in the assembly of iron-sulfur complexes in many of the repair proteins. It's really just a lovely breadth of work that Simon is doing, and I'm really looking forward to hear his seminar after we were able to enjoy a lot of time together yesterday. So Simon, welcome. It's great to have you here.
Thank you, Wolf. Well, it's a great pleasure to be here. I've heard a huge amount about the Davis campus from my good friends, Wolf and Steve and Neil, and it's been a real pleasure to be here. And I thank the Storers for supporting this lecture. It's very good of you to come, and hopefully I'll try and make it relatively straightforward for you to understand. But what I want to tell you about is how the process we use in my laboratory to really exploit the experimental strengths of a number of different model systems to try and improve our understanding of genome stability and how this impacts on human disease. And so it's a bit of a grand title, but hopefully at the end you will have got an idea about how we can use the power of genetics in the worm, and how we've used this as a kind of a starting point to discover new things which have extended to vertebrate models and then ultimately to human disease. So the starting point for what I've been interested throughout my career is genome stability, and even Google actually has cottoned onto this. You can get all sorts of amazing images, and of course this is DNA being blown apart by some kind of DNA damage. And here's another one. I think this is kind of nice. It illustrates the problem. We've got some kind of ray here blowing DNA apart. And really the point I want to make here is, I'm glad we've got this, I can walk around a bit, is that DNA is intrinsically unstable. It's prone to chemical change. Actually, the very active metabolism generates chemicals and reactive species that cause problems in DNA. And this is kind of simplified on this kind of rather generic slide that lots of people use, but it illustrates some important points. And that is that DNA can be modified and changed in many different ways. And this is really the challenge. So reactive oxygen species, these are kind of products of metabolism. They can react with DNA, generating base damage or single-strand breaks. Damage that will be familiar to all of you, of course, is UV. We don't have such a problem in the UK, but obviously you do in Davis with the kind of sun that you get here. But UV light causes changes in DNA. For example, thymine dimers, these are two T's next to each other. In the presence of, well, UV light can stimulate alterations to generate thymine dimers or 6,4-photoproducts. We actually exploit DNA damage, of course, in radio and chemotherapy. The idea is we want to damage the DNA to a point where it's irreparable and the cells die. And really, this is the challenge of the cell. These changes are happening all the time in our cells. Unfortunately, though, we have evolved very specialist and very effective ways of dealing with DNA damage in our cells. And what I've really been focused on throughout my career is how one particular type of DNA damage is sensed and repaired, and that is a double-strand break. So you could argue this is one of the most catastrophic forms of DNA damage, because of course you're breaking both strands of the duplex. So if you don't repair the damage, you're going to lose genetic information when you undergo cell division, for example. So I just want to tell you a little bit more about double-strand breaks. Of course, these are induced by ionizing radiation used in chemotherapy, as I've mentioned, and these are kind of what you could classify as spontaneous double-strand breaks. But actually, the very process of DNA replication, so copying your DNA in order to pass it on to daughter cells, is arguably the most relevant source of endogenous DNA damage, and this arises as a result of collapsed replication forks. But double-strand breaks aren't all bad. Neil will tell you we wouldn't exist without double-strand breaks. They're absolutely essential for meiosis and generating viable gametes for sexual reproduction during meiotic recombination. None of us would have a functional immune system. So the process of VDJ recombination, which is basically establishing a repertoire of antibodies, is completely dependent on double-strand breaks, programmed events in cells. But there's another class of double-strand breaks, and that is our chromosomes, or most species' chromosomes, are linear. You've got an end, and that end could be seen as a double-strand break. So what cells have evolved are these specialized structures called telomeres, and you'll hear a lot more about this. These are specialized structures at the end of all linear chromosomes that basically mask the end from being seen as a double-strand break. Okay? So you could class this as a masking of double-strand breaks. So you could argue there are at least 3 classes of double-strand breaks that the cell sees. So why do we study this? Well, it's intimately linked with human disease. So this is not an exhaustive list by any means, but there are lots of syndromes, disease syndromes in human beings that are associated with problems in repairing double-strand breaks. Very famous ones, ataxia telangiectasia. This is mutated in the kinase that recognizes double-strand breaks and signals to the cell. Severe combined immunodeficiency. These patients have no immune system. They can't make the antibody repertoire by VDJ recombination. And then we have other problems such as Werner syndrome and dyskeratosis congenita, and you'll hear a little bit more about that towards the end. And this relates to problems in telomere biology. Okay, so I'm going to give you a very simplistic overview. It's not entirely accurate, but it just illustrates a few points. And this is how double-strand break repair mechanisms differ in different stages of the cell cycle. So there are 2 major pathways of double-strand break repair that we know about in cells, and they operate in different cell cycle phases. So in G1, in the gap phase 1 of the cell cycle, if you get a double-strand break in DNA here, generally you use an error-prone mechanism of repair called non-homologous end joining. Essentially, you just stick the break back together. It's kind of a gluing mechanism. But the problem with this mechanism is completely independent of DNA sequence. So there is a propensity that if you've got a double-strand break here and another double-strand break in another part of the genome, because this is independent of sequence, occasionally you stick the wrong ends together. And this gives rise to translocations in cancer, for example, and you can see that. So it's error-prone because it's independent of sequence, and you can see the harlequin chromosomes that you can see here. You can see this is actually a translocation between chromosome 17, 16, and the X chromosome. And this almost certainly arose as a result of the inappropriate or misrepair of double-strand breaks through end joining. So this is the situation in G1, and I should say that end joining operates throughout the cell cycle. But once you've copied your DNA during DNA replication, you now have a template, an intact template through which you can repair. So you have this second choice. So once you're in SG2, you could repair this double-strand break using the intact sister as a repair template. And this is a kind of a photocopying mechanism. You're basically copying the sequence from here to repair the break here. And this is homologous recombination. It's slightly more complicated mechanistically than end joining, but essentially it's this photocopying mechanism. So absolutely essential for recombination is the RAD51 protein, and Raquel and Wolf and Steve Kolsikowski have worked on this for many, many years. And this is just a few important points about RAD51. So it combines to both double-stranded and single-stranded DNA and forms these long filaments. And this is an electron micrograph just showing you what it looked like. And we have crystal structures of this, sort of giving us really a detailed molecular insight into how RAD51 is working. Really beautiful work from Nikola Pavletic recently on how all of this comes together. So what RAD51 can do is really quite amazing. So it's loaded onto single-stranded DNA on processed double-strand breaks. And it has this amazing ability to find and engage with the homologous or intact template duplex DNA. So this then undergoes a transition with the displacement of the non-complementary strand. And so you then form this displacement loop intermediate, or D-loop. And this is going to be a major focus of what I'm going to tell you about. And once you've freed the 3' end, you can now engage polymerases to then extend this, to then copy the information. And then through some fancy gymnastics, you can repair the break. So this is really just a cartoon of what this is like. You've processed the double-strand break. This is then invaded into the homologous duplex DNA, and then you prime repair synthesis, and this is what happens, or it should happen. Okay, so you engage here, you form the displacement loop, you synthesize the DNA, and then you form basically intermediates that you will then process using specialist scissors, you could call them. These are nucleases that, dependent on the way you process these intermediates, can give rise to exchange or non-exchange of flanking DNA sequences, giving crossovers or non-crossover outcomes. So Wolf mentioned this. So one of the systems we use in my lab is worms, and they're quite amazing little things. And they have some really unique properties which we try to exploit in the lab. So they have a relatively short life cycle. The great thing about them is they're quite cheap to use. So you actually grow them on a lawn of E. coli at room temperature. So it's relatively accessible to most people. The great thing is, unlike Drosophila, I apologize to the fly people here, but we can store these indefinitely at minus 80 degrees, and we can defrost them 40 years later, and they start growing again. It's quite amazing. We have, there are 959 cells in the animal, and we have an entire cell fate map. But the thing that's so important to us about C. elegans is the genetics. It's a very robust and powerful system for doing genetic screens, and this is what we use the system for. But like any system, it has its limitations. We can't do proteomics and look at protein-protein interactions very easily in the worm. Doing structure-function studies, mechanistic studies are very difficult. This is now changing. There's some new technologies that are allowing us to do this kind of thing. But this is still somewhat tricky in worms, and because we work on a lot of disease processes, it becomes relevant to move it into more vertebrate systems. So we develop mouse models based on the C. elegans work we've done, and we do cell-based assays, as you'll see, looking at protein-protein interactions using proteomics and biochemistry as well to try and understand the proteins at the molecular level. So what I'm going to tell you about is a series of updates and stories on one of the proteins that we've been working on for a number of years that plays an important role in controlling recombination. So there are many proteins that have been implicated in executing recombination in a positive way. So proteins that play a role in actually processing the double-strand breaks to generate the substrate for recombination. This is called double-strand break resection. You've heard a little bit about the proteins that promote the invasion of the broken end into the homologous duplex. And this is Ra51. And proteins that may be very familiar to you are the BRCA genes. These are the hereditary breast and ovarian cancer tumor suppressor genes, BRCA1 and BRCA2, and if you inherit a mutation in these, you have about a 70% lifetime risk of developing breast or ovarian cancer. And the reason being is that these are absolutely essential for recombination. BRCA1 seems to play a role in the regulation of the break processing, and BRCA2 works intimately with Ra51 to promote the strand invasion event. So then you have various other enzymes. These are these specialist scissors I've mentioned, nucleases here and here, and also helicases and topoisomerases that can be differentially utilized to process these to give rise to different outcomes. But these are the things that positively influence the pathway. And what is true in all aspects of biology is that pathways need to be reversed under certain circumstances. So you have both positive and negative regulators of a pathway. And we became very much interested in how recombination was controlled from a negative point of view. And the starting point for the work that I will tell you about is this protein called SRS2. There's a very large body of work in budding and fission yeast describing SRS2 as a regulator of recombination. And this really was the starting point for the work that I'm going to tell you about. So what SRS2 does is it's recruited to sites of stalled replication forks, and the way it's recruited is through a dual interaction between PCNA and the SUMO form of PCNA, which there's actually binding sites for both of these moieties on SRS2. And so this protein, SRS2, was found over 30 years ago as a suppressor of Rad6. And the key features of this particular helicase is that the mutants are hyperrec. Basically, when you knock the gene out, recombination goes ballistic. The cells start basically doing recombination inappropriately. The cells are exquisitely sensitive to DNA damage. And biochemically, the way SRS2 works is essentially like a snowplow. It basically can act to drive the Ra51 protein from that filament that I showed you about earlier. So it can basically act to prevent the formation of the Ra51 filament on DNA. And this is believed to be how it works. So the important point here is that although there's a lot of literature on SRS2 in budding and fission yeast, there are no obvious functional homologs outside of yeast. So if this protein is so important in yeast for genome stability and for controlling recombination, who's doing it in us? That's the question. So we set out to do a genetic screen in worms, and the screen was based on the following observation in yeast, and that is that when you combine the SRS2 mutation with another helicase called SGS1, which is the Bloom's homolog in yeast, this kills kills yeast. This is a synthetic lethal interaction, and SRS2 is also synthetic sick or lethal with MARSEDI1. What is remarkable about this is that if you knock out Ra51, that recombination protein, you completely rescue the lethality of these mutants. So this tells you genetically that the synthetic lethality of these double mutants is due to the accumulation of toxic recombination intermediates. So what we set out to do was to use the worm. Now, sorry about the names. As you probably know, names differ in different species. HIM6 is the SGS1 or Bloom's homolog in C. elegans. So what we did was to do a genetic screen to try and identify genes that were synthetic lethal with the SGS1 or Bloom's homolog in worms, with the view to trying to identify something that was functionally analogous to SRS2. To cut a long story short, we discovered a gene called RTEL. So this is just showing you the synthetic phenotype. So we have, just looking at progeny viability, you see 100% progeny viability in the wild type. It's slightly reduced in the RTEL single mutant. There's a role for HIM6 in crossover formation and meiosis, and this accounts for the reduced viability that you see here. But when you combine both mutations, you can see this synthetic phenotype. So this is now over 90% of the progeny die in the absence of both of these two helicases. So what we went on to show is that in addition to synthetic lethality between HIM6 and RTEL, we also identified 3 other synthetic lethal interactions between RTEL and RECQ5. So double mutants between RTEL and RECQ5 are synthetic lethal. The double mutant of RTEL with MOS81 is synthetic lethal, again, analogous to the situation in yeast. And a gene called DOG1, which is actually the C. elegans homologue of the Fanconi anemia mutated gene FANCJ, is also actually cell lethal in worms. And what we could show, that, and again analogous to the situation with SRS2 in yeast, that this lethality in all 4 cases was due to basically excessive recombination. So these worms were dying with very high levels of recombination intermediates. So this was very interesting to us. We started to study the RTEL single mutant. It has reduced bioavailability, as I've mentioned. It has exquisite sensitivity to DNA damaging agents, and it has a hyper-rec phenotype. So when we look at recombination in the single mutant, it's elevated. So that's the C. elegans situation. What we found is also a human equivalent of this gene, and we could show, at least for damage sensitivity and the recombination phenotype, this is also true if you downregulate the RTEL gene in mammalian cells. So these phenotypes, so the synthetic lethality and excessive recombination and the hyper-rec phenotype, were all consistent with a role for RTEL in constraining recombination. So what we did was to go on to try and understand how this worked biochemically. So we purified the proteins, and we first of all asked whether or not it worked in the same way as SRS2. So just to remind you, what SRS2 does is it strips Rad51 from the single-stranded DNA at the process double-strand break. So we can reconstitute this with purified proteins. So if you take radiolabeled single-stranded DNA, you then add Rad51 and RPA, you can form this nucleoprotein filament, and I showed you the EM image of that, and you can see this retarded species on the gel. This is the Rad51 nucleoprotein filament. What we asked is what happens when you add the RTEL protein? Does this dismantle the filament? Well, you can see that it has really no measurable impact on the stability of the filament. We're adding increasing concentrations of the wild-type RTEL protein, and there's no measurable change in this filament. So in contrast to SRS2, it has no measurable impact on the stability of the Rad51 nucleoprotein filament on single-stranded DNA. So we switched our focus to another intermediate in the recombination process, and that is the D loop that I mentioned at the beginning. And so this is the point where the broken end of the molecule, the double-strand break, is invaded into the homologous duplex DNA. So again, using purified proteins, what we can do is pre-form D loops in vitro. So in this experiment, we've got radiolabeled single-stranded DNA. We then incubate that in the presence of Rad51 and RPA with a supercoiled plasmid that contains homology to the single-stranded DNA. And Rad51 catalyzes the invasion of that labeled single-stranded DNA into the supercoiled plasmid. So this is a D loop. So we pre-form the D loop, and then we ask what happens when we add the RTEL protein. And what it does is it beautifully disassembles the D loop. Okay, so it takes the D loop apart. And this is absolutely dependent on the ability, on the ATPase activity of the RTEL helicase, because if we make a point mutation in the Walker A box, this completely kills its ability to disrupt the D loop. So then simplistically, what RTEL is doing is it's acting on this intermediate here to reverse the process. So this is how we think it's working as an anti-recombinase. It's reversing the process. So what we've been doing over 4 or 5 years is trying to understand how RTEL works in D-loop metabolism. Now, of course, we can show this in vitro. The question is, does it actually happen in vivo? And this is a challenge that we have, and pretty much any biochemist has. So what we've been doing is looking in different physiological contexts during DNA repair, DNA replication, in meiosis, and at telomeres. And these are just coming back to what I introduced at the beginning. These are all places where the D-loop intermediate is is believed to play an important role in the outcome of these different events. So how does RTEL work in D-loop metabolism? So what we basically have accumulated over the years is I think we have a body of evidence now that is consistent with RTEL working at a D-loop. We can't say for definite, but I think it's consistent. So Wolf mentioned we're also very much interested in how RTEL is regulated, of course, and Wolf actually mentioned this work that we've recently published describing how RTEL and many other DNA repair and DNA metabolism enzymes, actually their activity is strictly dependent on the assembly of an iron sulfur cluster, and we provided some new insights into how this occurs. So we think RTEL plays an important role in counteracting toxic recombination, and this was consistent with some of our published work, the early work on RTEL. And we also have some very interesting unpublished work that is, again, consistent with RTEL counteracting toxic or illegitimate recombination in both C. elegans and in mouse cells. So several years ago, we also have analyzed the role of RTEL in C. elegans meiosis, and the take-home message of this was that in the RTEL mutants, we actually get much higher levels of crossing over, which is above and beyond what you see in the wild type. And so this was really consistent with the idea that RTEL is promoting a non-crossover outcome of repair, and again, in various models, this implicates a requirement for D-loop metabolism to execute this process. But what I want to tell you about today is work that we recently published relating to the telomere function of RTEL, which again invokes the need for a D-loop, as you'll see, a D-loop metabolism enzyme in this process. And I also want to go on and tell you about some very exciting new unpublished work that really begin to give us an indication as to how these activities of RTEL are executed at the telomere. So RTEL, why is it called RTEL? Well, actually, it was work from Peter Lansdorf in 2004 that first described RTEL as a regulator of telomere length maintenance, hence RTEL. So he discovered this in a really complicated screen to look for telomere, the factors that influence telomere length between different mouse species. And basically, using complicated mouse genetics, he mapped the loci or the locus that's responsible for these telomere length differences to the RTEL locus. So he knocked this out in mouse. The mice are dead. And this actually, he has no idea why RTEL is important for this difference. But really, what took our interest was the fact that when you derive cells from these mice, you see really remarkable genome instability. You see lots of chromatid breaks. You see a very dramatic telomere phenotype, which I'll show you in a minute. It's really striking. What wasn't apparent at this, in Peter's original work was that telomeres are very fragile, and I'll again tell you what that means in a minute. And this only really became evident as a phenotype linked to RTEL from work from Tisha de Lange in 2009. So it has this remarkable genome stability phenotype, particularly a very important role in regulating telomeres. But we have no idea how it functions at telomeres, and this is really what we were interested in trying to understand. So what are telomeres? Let me just remind you. These are these specialized structures that basically protect and regulate the end of linear chromosomes. So they're not really red. This is just showing you where they are. They're at the end, okay? So what are telomeres? Well, they're actually in humans and actually other species, they're generally a repetitive sequence. The human sequence is TTA, GGG, and this is repeated many, many times. Human telomeres are about 10 to 20 kb in length, made up of this repetitive sequence. And you can see that, in fact, you have both a double-stranded telomere sequence and a 3' single-strand overhang. So you have both double and single-stranded DNA. So this whole repetitive sequence at the end of the chromosome is bound by very specialist proteins. Two of them, TRF1 and TRF2, they bind specifically to the double-stranded telomere repeat sequence, whereas POT1 binds to the single-stranded DNA at the end of the telomere. And together, these are all linked together by several other proteins, TIN2, TPP1, and also RAT1, which isn't on here. And together, these form a complex, which is called sheltering, because it shelters the chromosome end from being seen as a double-strand break. So what this this complex does is it regulates and recruits telomerase. So this is a specialized enzyme that helps to extend the telomere repeats. And this is because semi-conservative replication can't replicate all the way to the end. This is called the end replication problem. So you need to maintain the telomere, and you do this by telomerase. As I've mentioned, this whole structure protects the chromosome end from being seen as a double-strand break. And also there's emerging evidence that actually this complex helps the replication of the telomere, probably because you form a lot of secondary structures from these repetitive sequences, and the binding of these proteins help to minimize these sequences to help replicate the telomere. Okay, so where we became very interested in this was really prompted by this. This was actually the front cover of Cell in 1999, and this again was work from Tisha DeLange and Jack Griffiths. And what you can see is a lasso-like structure. So this is an electron micrograph of a telomere from human cells. And this was really provocative at the time, and since then there's been a lot of evidence to suggest that what this structure is a recombination intermediate, where the 3' end of the telomere basically reinvades or invades into a double-strand telomere repeat internally to form this structure. And what's nice about this is it protects the end. Remember, this is all bound by the sheltering proteins as well, but it's like you've capped the end, you've hidden the end from being seen as a double-strand break. So in that respect, it's quite an attractive model. But I have to say it's a little bit controversial in the field, principally because it's almost impossible to show that this structure forms at every telomere, and that's the problem. But I'm sure all of you here will be familiar with this structure already, because what this is a D-loop. Okay, it's the D-loop that I've been telling you about. So we had this idea, and that is that we have good evidence genetically and biochemically that RTEL can reverse the recombination reaction of a conventional D-loop to basically counteract toxic recombination, for example. The question is the telomere phenotype of RTEL deficient cells a reflection of a problem in basically dismantling this structure? And that was really what we set out to test. So the way we did this, we had to do this in mouse cells. We basically developed a conditional mouse knockout cell that would allow us a window of opportunity to look at what happens when you inactivate RTEL and then look at it over time. And so the way we do this is to basically use specialist technology where we can flank the RTEL gene with LOXP sites. We then add Cre recombinase expressed from a virus. And this is now a Western blot, an IP Western blot of RTEL, 96 hours after we've infected these conditional cells with the Cre recombinase. And you can see that we lose the protein. So this is then we can then study what happens to the cells. I'm not sure if you can see this, but I'll give you zoom. This is a metaphase spread of RTEL deficient cells 96 hours after Cre. So you've lost the protein. And what you see are these remarkable telomere phenotypes. So what we're able to do is to visualize the telomere by telomere repeat fish. Okay, so these green dots are the telomeres. Okay, and what you can see straight away is that these telomere signals differ in intensity. So this actually is a reflection of telomere length changes. This is actually telomere shortening, but you will also notice this very bright spot here. This is actually telomere lengthening. And so actually, if we look at the frequency of recombination of the telomere, it goes up dramatically when you get rid of RTEL. And this accounts for basically an establishment of a new telomere length homeostasis. So telomeres become very heterogeneous in length. We also see really dramatic events. And that is where we completely lose the telomere. As you can see here, we've completely lost the telomere, and this we see a very high frequency in the metaphases as well. So this is absolutely remarkable. So people who are familiar with the telomerase knockout mouse, it took 6 generations before you started to see a problem. So that is that the telomere got progressively shorter to a point where you hit catastrophe. You couldn't maintain the chromosome end without the telomere, and it took 6 generations. This is happening in a single cell cycle. So something really dramatic is happening. So we also see telomere fragility. So what is this? Well, you're probably familiar with the term common fragile sites. These are intrinsically unstable regions of the genome. They are Particularly, they're induced if you start slowing DNA replication. Well, telomere fragility is basically common fragile sites at telomeres. So this phenotype, as you'll see, gets much worse if you start slowing DNA replication. Why you start to see these multiple telomere fish signals, which is basically how this phenotype manifests, we don't know. But this is basically telomere fragility. So why are we seeing this in the RTL deficient cells? So we set out to test whether this was a problem in controlling recombination or potentially dismantling the T-loop structure. So this was the question. How do you go from this to this within a single cell cycle? Well, it's unlikely it's degradation based on the telomerase knockout. So what we hypothesized is, in fact, what was happening is that this structure was being erroneously processed by nucleases, these specialist scissors, giving rise to telomere circles. Now, this wasn't completely fictitious, because in fact telomere circles have been described in the telomere literature before, for example in alt cells which maintain telomeres in the absence of telomerase by recombination, and also in various dominant negative experiments. TRF2 delta B cells overexpressing this dominant negative produce very high rates of telomere circles. So this was of course a model that we had to test. So what we wanted to ask are, when you knock out RTL, do you actually induce the formation of telomere circles, which would be consistent with this model? And if so, who is responsible for cutting the telomere off as a circle? And that's what I'm going to show you. So how do we detect telomere circles? So this is the assay. It's adapted from a method first described in plants. It's really quite straightforward. So you take genomic DNA from the cell, you denature it, you then anneal a telomere-specific oligo, and then you use a rolling circle 529 polymerase to amplify up the signal, okay? And the way you can distinguish between linear and circular molecules is by adding XO5. So XO5 needs an N to degrade, so circles are resistant, which you can see here. So basically the circles, when you run them out on a gel, run high up in the gel. These are the linear telomere fragments that when you treat with XO5, they disappear. So these are circles, okay? So we can use this assay in the RTL knockout cells. So this is just one experiment. And we're using VA13 cells, which are an alt cell line, as a positive control. And you can see that we accumulate nice levels of telomere circles in the VA13 cells. So we see a background level of telomere circles in the RTL floxed over minor cells, but when we add Cre to get rid of the RTL, remaining RTL gene, we see this very rapid accumulation of telomere circles. Okay, so this is all happening within 72, 96 hours. So this is just a 96-hour time point. But we've done time courses of this, and in contrast to the heterozygous situation, where you don't see any accumulation of telomere circles, in the RTL knockout cells, we see this rapid accumulation of telomere circles. So these are occurring very, very rapidly, almost coincident with the loss of the telomere or the change in telomere length. So, I mean, this is telling you something that took us 3 years to work out. So I'm skipping a lot of stuff, but we spent a lot of time thinking about who this pair of scissors would be. Well, to cut a long story short, we thought it was MUS81. We made the double knockout mouse, no phenotype. It was a killer. It took us a year, and it was a negative result. Anyway, it wasn't so bad because Jennifer Svensson, who worked with Wade Harper, joined my lab around the time that this paper came out describing SLX4. So SLX4 is a scaffold for 3 structure-specific nucleases, MUS81, SLX1, and XPF. And this was really attractive, and when Jennifer came to the lab, she said, oh, well, you know, why not look at SLX4? There's a very good reason why this might be the nuclease that you've been looking for. And the reason is that not only does this nuclease complex have activity towards this kind of substrate, it is also known to bind to telomeres. So in the proteomics of the SLX4 complex that Jennifer described, you could see TRF2 and RAP1, 2 of the sheltering components. So this enzyme was ideal, okay? So this is just showing you the interaction between SLX4 and TRF2, one of the sheltering components. And this is just showing you that actually SLX4, labeled with GFP here, is largely coincident with RAP1, one of the sheltering components. So this nuclease complex has the right activity to process the T-loops and is at the telomere. So it's at the right place and has the right activity. So this was actually the first experiment we've now confirmed this in double knockout mice. But basically what we've done is to take those RTEL floxed over minus cells. We've then stably expressed either a control hairpin or the SLX4 hairpin, and we've done this with multiple, so we've excluded off-target effects. And what you see is that when you knock down SLX4, you completely suppress the telomere circles. Okay? So the formation of those circles is dependent on SLX4. Now, what I've been kind of alluding to you all the way along is that the loss of the telomere, the telomere circles are coincident and actually responsible for telomere loss. If that's the case, then we should suppress telomere loss by downregulating SLX4. And this is the experiment. And it was beautiful when we did it. So this is now showing you the number of telomeres lost per metaphase. Okay? This is now the RTEL floxed over minor cells before Cre, so these are kind of wild type. And you can see that there's no measurable effect of downregulating SLX4 with two different hairpins. You then add Cre to the cells. 96 hours after Cre infection, you've lost RTEL, and you accumulate very high levels of telomere loss in any one metaphase. So what happens when you downregulate SLX4? You completely suppress this phenotype. So you suppress telomere circles and telomere loss. And this is completely independent of cell cycle. Okay, so coming back to this other phenotype that I mentioned at the beginning, telomere fragility. Well, this was really bizarre because actually when we looked at telomere fragility in this experiment, it was SLX4 independent. Okay, so here we're looking at the number of fragile telomeres per metaphase, and it basically doesn't change when you knock out SLX4. And this was the first indication that maybe there are two roles for RTEL telomeres. One is suppressing this telomere length changes, which is SLX4 dependent, and fragility seemed to be somewhat different. So this is really a model that was taken from Tisha DeLange's lab. So what they proposed is that, in fact, TRF1, one of the sheltering components, was facilitating replication through the telomere by suppressing the formation of G quadruplex structures, secondary structures, formed by these repetitive sequences. Now, this is what she proposed, and there was no formal evidence to say that this was actually the case. There is clearly a replication defect through the telomere in the absence of TRF1, but evidence that G quadruplexes are a problem was not shown in this study. So what we did is that we took a drug, TMP-YP4, and this is known to bind to and stabilize G quadruplexes. And we thought, well, if G quadruplexes are a source of telomere fragility in cells, if we stabilize them, we should make the phenotype worse. Okay? So this is now just showing you telomere loss per metaphase. And you can see that adding TMP-YP4 has no measurable effect on telomere loss. Okay? So these are is independent of G quadruplex stabilization. However, when we look at telomere fragility per metaphase, what you can see is the telomere fragility that you see when you knock out RTEL. Okay? Then you add G quadruplex stabilization, drug, TMP-YP4, and this phenotype gets much, much worse. Okay? So what this suggests is that at least a large proportion of the fragility, the problem in replicating through the telomere, reflects a problem in unwinding secondary structures, such as G quadruplexes, formed by the telomere repeats. So this is just summarizing what I've told you. So what we think is that there are actually two functions for RTEL that reflect the changes in telomere length, telomere loss, and telomere shortening, and a second role in suppressing telomere fragility. So what I've shown you is that telomere loss happens coincidentally with the rapid accumulation of telomere circles. So what we think is that basically the telomere is being processed inappropriately as a circle, and that loss of the circle gives rise to the loss of the telomere sequence. And what I've told you is that the telomere circle and telomere loss is actually catalyzed by a nuclease that is resident at the telomere, the SLX1/4 nuclease complex. And this is unaffected by G quadruplex stabilization, and data I didn't show you is that actually we can completely rescue this phenotype simply by slowing DNA replication. So we think that the excision of the telomere as a circle requires active DNA replication, probably the collision of the replosome with this structure. Conversely, telomere fragility is quite different and is genetically separable. I'm going to further convince you of that in the next little part of the talk. So in fact, telomere fragility, and this was shown by Tisha DeLange, is actually exacerbated by inhibiting DNA replication. So if you add a low dose of afidicolin to levels normally used to induce fragile site expression, this phenotype gets much worse, and it's obviously directly opposite to the telomere loss phenotype. It's independent of SLX4 and gets much worse if you stabilize G quadruplexes. Okay. So this is kind of a model of what we think is going on. In normal cells, somehow RTEL is recruited to the telomere. It uses its D-loop disrupting activity to unwind the T-loop. And this has two functions. It allows the replisome to complete semi-conservative replication. It also allows telomerase access to the end to solve the M replication problem. Okay? And this was all based on observations we made in the RTEL deficient cells, that when you don't have RTEL, what happens is that this persistent T-loop is processed by the SLX4 complex, giving rise to a crossover, which would produce a circle, and this then gives rise to telomere loss. So that's kind of what I've shown you in a model form. So what we also have evidence for is the second role for RTEL in suppressing or unwinding telomere G quadruplex forming structures. This is also a function that's shared with the Bloom's helicase, and in fact, if we make double knockout mouse cells of these two, the phenotype gets much worse, suggesting that they're operating in different pathways. So this is kind of what we're proposing. There are two roles for RTEL at telomeres. It disassembles T-loops, and this is important for preventing the catastrophic processing of the telomere by the SLX4 complex. And there's also a separable and distinct role for RTEL in counteracting telomeric G4 DNA structures to help with replication through the telomere. And I'm not going to show you the data because I don't have time, but we have biochemical evidence for both of these. RTEL will unwind T-loops formed in vitro, and it will unwind G quadruplexes. And this is actually inhibited by adding TMP-YP4. So this is kind of where the paper ended. What we've become very much interested in is how are these two different roles executed at the telomere, and that is what I'm going to tell you about now. Okay, I'm going to have a drink. I'm getting a bit parched. So what we've been looking at is trying to understand how the RTEL protein works in all of this. And of course, it has the helicase domain at the N-terminus. And what we noticed very early on is that it has a beautiful pip box. So what a pip box is a PCNA interaction module. And I'll just show you the biochemistry, then I'll tell you what PCNA is. So we can see the pip box dependent interaction with PCNA in multiple different assays. This is a V5 knock-in ES cell line expressing a tagged form of RTEL from the endogenous locus. We pull down RTEL with a V5 tag. We pull down PCNA. We can make a GST fusion to this part of the RTEL protein contained in the pip box. If we make a single isoleucine to alanine substitution, you can see that the wild-type GST protein can pull down recombinant protein or PCNA from two different cell lines, but point mutation in the pip box kills this interaction. We can also spot biotinylated peptides onto nitrocellulose membranes, either the wild-type sequence or the pip box, the I2A substitution change here. We can then incubate with recombinant PCNA, wash, and then Western blot, and you can see that only the wild-type pip box containing sequences bind to PCNA. So what this tells you very quickly is that RTEL binds to PCNA, and it does so through this pip box. Okay. So what is PCNA? Well, it's the processivity factor for DNA polymerase. It's absolutely essential for DNA replication, but it's also implicated in DNA repair and DNA damage tolerance. And this is this protein has received a lot of interest in the field for many years. So unfortunately, we can't overexpress RTEL. It kills the cell, so we can't do the standard transfection of mutants. So we had to take the plunge and do the experiment properly. And the way we did that was to make knock-in mice. So we basically knocked in this isoleucine to alanine change into ES cells and used these to generate mice. But as I mentioned, we also introduced a V5 tag at the C-terminus in order for us to follow the protein, because actually the levels are so low, we have to do IP Westerns to see it. So this is just the strategy to knock in the pip box mutation. The only thing that's really important on this slide is this experiment here, where we're IPing the wild-type RTEL. These are MEFs derived from the mice that I'll tell you about. This is the wild-type with a V5 tag. You pull RTEL down, you pull down PCNA. Whereas the single isoleucine to alanine substitution kills the PCNA interaction in vivo. Okay. So what happens? This is a single amino acid substitution, and so I'm showing you primary knockout MEFs or knock-in MEFs for this pit box versus the wild type control cells from the same breeding litter. So what we see are a number of phenotypes. We see reduced cell growth of this cell line relative to the wild type. We see increased senescence. These rapidly senesce, these cells, and we see decreased BRDU incorporation. So this was all really suggestive of a problem in DNA replication, and this kind of fitted with PCNA being the processivity clamp for polymerase. So what we've done is to look at DNA replication fork rates and dynamics using DNA combing methods, where we basically stretch out the DNA after we've labeled active DNA replication with either iodo or chlorodeoxyuridine, so that we can then distinguish between these two different analogs with different antibodies. And all of the experiments are done in primary MEFs under low ambient oxygen conditions. This is to try and minimize oxidative damage. And what you see, and I hope you can see this at the back, I've shown you the quantification underneath, is that very similar to the RTEL null cells, the pit box knock-in cells have much slower DNA replication fork rates. This is just showing you an example of a wild type fork. This is an origin here. You have the extension that you see with the iododeoxyuridine. You then switch to chloro, and you can see the length of the track. So you can see in the RTEL pit box knock-in cell that the track lengths are much, much shorter. And this tells you that replication fork rates are slower in these cells. And we're not damaging them. This is just under normal replicating conditions. And basically what you can see here, this is now basically the iododeoxyuridine replication extension rates over multiple DNA combing fibers. And you can see that the fork rates are dramatically reduced relative to the wild type. And we see a similar kind of trend in the null cells, but we can't directly compare these because they're completely independently derived cells. I hope you can see this at the back. So in addition, from these combing experiments, we can also get an idea of the prevalence of fork stalling or collapses. And that's by looking at the asymmetry of the fork. So you can see this in a minute. So basically in the RTEL proficient cells, you can see that the lengths of the iodo and chlorodeoxyuridine tracks are very similar between different experiments. However, what you can see in the pit box cells is that there is a huge amount of asymmetry. So for example, here you have a red track here of reasonable length, and then the green, you have the origin here. You see the red track here is suddenly stopped. Okay? This is consistent with a stalling event or a fork collapse event. And this happens at a very high frequency. This is just showing you the quantification. This is the percent asymmetry. Now, it's a little bit confusing. You think, how can you have over 100%? Well, what we're saying is that a difference of 100% in a particular replication fork is simply a twofold change in the length. So if you have a fork that measures 1 centimeter, which of course it doesn't, and another fork that's 2 centimeters, there's basically 100% asymmetry. But what you can basically see in these experiments is very similar to the complete knockout of RTEL. The pit box knock-in cells have a very high frequency of asymmetric forks relative to their wild-type controls. Okay. We can also see this in a different way. So if we plot the length of the left moving fork against the right moving fork, okay, and basically you would expect that if the fork was completely symmetrical with no problems, basically everything should lie on this straight line. Okay, but if we divide this graph into this quadrant here, and then look at the number of forks that lie outside this will give us a measure of fork asymmetry. Okay, what you can see is that when we do this with the wild-type cells, most of the forks, over 90%, lie within this linear range. About 7.8% are outside. So there's a 7.8% of the forks in the wild type are somewhat asymmetrical. However, you do the same analysis in the pit box mutant, you can see that over 50% of the forks lie outside this range. Again, an indicator of fork asymmetry. So more fork stalling or collapse. So how do cells survive? So if you've got slower fork rates, the forks are collapsing, how do the cells survive? Because they still grow. And the way they do that is to basically start firing dormant origins. So you start basically compensating for this problem in replication by are basically firing extra origins. And so an indirect measure of that is look at the inter-origin distance. So here we have a replication origin here, here, and here in the wild-type cell. And so we can then measure the inter-origin distances here versus in the wild-type. And so we can quantify that here. So this is the inter-origin distance in the wild-type versus the pitbox mutant. You can see that there's quite a significant decrease in inter-origin distances in the pitbox knock-in. So we think that the way that the RTEL pitbox knock-in cells compensate for this replication problem is by increasing the usage of replication origins. So what about the telomere phenotype? This is what we were really interested in. So just to remind you, we see this telomere circle formation and loss by the SLX4 complex, and telomere replication problems associated with unwinding of telomere G quadruplex structures. So this is the experiment that really looked at one aspect of that, and that's telomere circles. So this is an experiment you've seen before. This is the wild-type RTEL deficient cell, and when we inactivate RTEL, we see this very high level of telomere circles. However, you can see straight away that, in fact, the pitbox mutant doesn't show any measurable difference in telomere circles relative to the wild-type cells. This is just showing you the quantification. So, as you might expect, there's no telomere circles in the pitbox. There's no measurable difference between the wild-type and the pitbox knock-in cells for telomere loss or telomere length changes. So this is telomere loss per metaphase, and as you've seen before, this is the wild-type. You knock out RTEL, you get very high rates of telomere loss, but you don't see this in the pitbox mutant. What happens to fragility? And this is kind of geneticist's dream. It's a complete separation of function. So basically what you see, this is now fragile telomeres per metaphase in the wild-type. In the null, this is the pitbox as wild-type V5 cell line, and this is the pitbox knock-in. It looks indistinguishable from the null situation. So this is a separation of function mutant. So the pitbox mutant exhibits telomere fragility without telomere circle formation or telomere loss. Okay, so we can add a little bit more information to the information I gave you from the first paper. And that is that the accumulation of telomere circles and telomere loss are independent of its ability to interact with PCNA, whereas telomere fragility, which is a reflection of a problem in replicating through the telomere, requires an interaction between RTEL and PCNA. And this kind of makes intuitive sense, I think. Okay. So I told you at the beginning that the RTEL null mice are dead, dying between days 10 and 11.5. Now, despite the telomere phenotype, the replication phenotypes, but are compensated by upregulation of origins, the knock-in mice are viable. Okay, so this allowed us to do some aging and tumor watch studies. So I'll just tell you part of the data that we have. So we looked at the impact of this single pitbox mutation on tumorigenesis. We kind of cheated a bit because these experiments can take 2 or 3 years. So we kind of cheated by knocking out P53, which accelerates tumorigenesis in many contexts. So what I'm showing you is the tumor-free survival in the P53 null with the pitbox mutant as a heterozygous, okay? And this is what you see. And many of the mice here die of lymphomas, which is basically a characteristic of P53 deficiency in mice. We then looked at the pitbox homozygous mutant in the P53 null background, and what you see is a really remarkable acceleration of tumorigenesis. We see many of these mice come down with lymphoma, but they also present with medulloblastoma. And this is really unusual. We're now starting to see a different spectrum of tumors simply with this single pitbox mutant change. We've begun to analyze this in a lot more detail, but I think what's really interesting here is that all of the telomeres that we've analyzed in the pitbox knock-in case exhibit very high levels of telomere fragility, which is what I've told you about. There's no obvious telomere loss, and you don't see a very high frequency of telomere fusions. And we never see these events in the heterozygous pitbox mutant case. So we could argue that this telomere fragility may be contributing to the genetic instability in these cells. Okay. So what have I told you? So RTL associates, we think, with PCNA and probably the replosome, and actually we have live cell imaging showing that actually RTL can associate with active replication forks in cells. And we believe that it's very important for facilitating replication. And this is based on the fact that replication fork rates and fork stability is profoundly affected simply by knocking out either the full-length RTL gene or preventing it from binding to the replosome. What we were very happy about is that we got this beautiful separation of function mutant. So by knocking out the interaction between RTL and the PIP box, we could show that the telomere loss catalyzed by the SLX4 complex is completely independent of RTL's ability to bind to PCNA. Whereas the ability to help replicate the telomere, probably in unwinding G4 structures, is completely dependent on the PIP box. So this is kind of the idea that this aspect of RTL function, namely helping replication of the telomere, is dependent on this PIP box interaction with the replosome through PCNA, and we think this arm helps with this particular feature of the RTL phenotype. So what about T-loop disassembly? Well, what I'm not going to show you is that actually we looked in the PIP box knocking cells, which had a V5 tag at the end, and we could actually see occasionally that PIP box mutant RTL still at the telomere. And this raised the possibility that RTL may also bind independently, potentially, to the sheltering complex. And so we went out to test the possibility that RTL may be recruited to telomeres independently of PCNA through an interaction with sheltering, and that's what I'm going to show you. So what we've looked at are each of the components of sheltering: TRF1, TRF2, RAT1, TIN2, TPP1, and POT1. And this experiment is just showing you negatives, namely that you IP RTL through a flag tag at the N-terminus, irrespective of the presence or absence of benzonase, which excludes DNA bridging. We do not see an interaction with POT1. Okay, so in this experiment, we're actually pulling down IPing POT1, and you don't see RTL. Same experiment, IPing TRF1, we don't pull down RTL. TPP1, no RTL. However, we then looked at TRF2, and this was beautiful. So basically, we IP TRF2, we pull down RTL, and this is resistant to benzonase treatment, suggesting it's not a non-specific interaction by DNA bridging. We can also see a weak interaction with RAP1. So if we IP RAP1, which is a binding partner of TRF2, we can see that TRF2 comes down in the IP, and we also pull down RTL as well. And actually, we think this interaction with RAP1 is indirect through TRF2, because if we downregulate TRF2, we lose the interaction between RAP1 and RTL. So I'm not going to show you that data. The important point is we can also show this. There is a direct interaction. For example, if we make a GST fusion to TRF2 or various different domains of mutants of TRF2, we can see that RTL binds to the full length, the TRF2 delta B form of RTL, but not to the delta B region alone. So we think this interaction is direct. I'm skipping through quite a lot of work just to tell you that we've mapped the interaction between TRF2 and RTL to what we've called TIM, for TRF2 interaction motif, and it's actually C-terminal to the PIP box. And what is really cool about this is when we looked at the sequence, it looks like this. It's a C4-C4 motif, okay? So this, we kind of thought maybe coordinating a metal ion or two, maybe. So first of all, we wanted to test, and I wanted to convince you that this domain is actually sufficient to bind to TRF2. So we made a GST fusion, and we performed a pull down from nuclear extracts from HEK293 cells, and this is the inputs. And you can see that GST does not pull down TRF2, but the GST TIM domain alone pulls down TRF2 beautifully. Okay? So is this dependent on metal coordination? Almost certainly yes, because if we include EDTA in the pull down, we completely kill the interaction with TRF2. We have no idea what the metal ion is at this point, but what we do have, and I don't have the data here, is that we can basically mutate some of these cysteines. We completely kill the interaction. Okay, so what's the phenotype? Now, we didn't make the knock-in mouse. We're doing those, but what we've been able to do is develop a retroviral complementation system of the RTL null cells. So we've knocked out the TIM motif. And what we get is the reciprocal separation of function. So what we see is telomere loss and telomere circle formation without telomere fragility. And I'll just show you some of the data. This is the RTL null cells corrected with the wild type RTL and the TIM mutant. And what you see is very high levels of telomere circles, and this is associated with telomere loss. So just to come back to the model, PCNA interaction is important for the replication of the telomere and unwinding of telomere G4 quadruplex forming sequences, whereas T-loop disassembly, we believe, is absolutely dependent on the ability of RTL to bind to TRF2 by this TIM motif at the C-terminus. And we think that this is very important for bringing RTL to the telomere to dismantle the T-loop. And we don't know how this is regulated at this point, but it's certainly dependent on this interaction. Okay, so I told you that well, the start of this whole story was genome stability from worms to human disease. So we started in worms, we've gone through the mouse, and in the last just minute I'll tell you that actually RTL is a very frequent mutation. Mutations in RTL are very common in glioma, okay? It's overexpressed in many cancers, such as in gastric cancers, and we actually have reasonable evidence now that we think this mutation is actually causing problems in controlling homologous recombination. And in fact, if you conduct basically a meta-analysis of RTL mutations or alterations across many of the different cancer data sets that are available, it is amplified at a very high level. And what I think is kind of interesting about these experiments, these cells, is that cells that overexpress RTL are extremely unstable genetically, and they're also sensitive to PARP inhibitors, which I think is kind of interesting. So the other disease link has come about through an interaction with good friends of a number of you guys in the audience, John Petrini at Sloan Kettering. John has discovered mutations in RTL in a syndrome, I'm going to try and say it, Hoyer-Horidason syndrome, okay? This is a very severe form of dyskeratosis congenita, and this is caused by mutations in telomerase components or in TIN2 and other sheltering components. So Hoyer-Horidason syndrome is caused by mutations in RTL. So unfortunately, this is a really horrendous disease. The children die about 3 or 4 years of age from cyclomegalovirus infection because they're immune deficient, but they also present with aplastic anemia, microcephaly, cerebral hyperplasia, and their growth retarded, okay? Well, what we're doing now is working with John using all of the tools that we've developed to look at RTL to try and understand why these particular mutations in RTL give rise to this particular disease. So what I want to do is just thank the people who've done the work. So everything I've told you about RTL and telomeres was done by this guy, Jean-Baptiste Vanier, fantastic postdoc in the lab. The interactions between RTL and the sheltering component were done by Greg Sarek. Louise Barber and Jill Utes were the people who did the genetic screen that first found RTL in worms. And Jill followed this up with the meiotic analysis of RTL, which we implicated in a role for RTL in non-crossover repair. These have had help from Vish Kaltenbrunner and Mark Petal-Corin in the lab. The pitbox knock-in mouse analysis was done in collaboration with Hal Ding, and you might remember that Hal was actually the first author on the original RTL paper from Peter Lansdorf's lab in 2004, and his student Zinatan Nabi. These guys, Tisha, John, Claire, and Alan, have provided us with lots of mouse strains, antibodies, and knockout ES cells. These are the people who fund our work, and although I'm at Clare Hall at the moment, we're going to be transferring to this Francis Crick Institute, which I think is a little bit more, a better name than this rather bland London Research Institute. Anyway, this building is going up at the moment. It's going to be a massive new institute right in the heart of London, so I think it's going to be exciting. And finally, this is all the lab at the moment. These are the guys who work with me, and they're a great group. And anyway, I'll thank you for your attention, and I'll take questions. Thank you.
Thank you, Simon, for a stunning talk. And now you know why we spent no time choosing him for this invitation. So the questions are open. Super.
So I was wondering, given the functional analysis you've done, is there any explanation of the basis of the different length of the telomeres in the two mice?
Yeah, I kind of defer to Peter here. I mean, he looked for it. He found artel as the locus responsible. I think there's no question that knocking out artel leads to changes in telomere length. But what is not clear is why the two different mouse strains actually differ. There's nothing obvious, like, for example, polymorphism differences between the two mouse strains. What they've not done is do a replacement, for example, putting the spretus artel into musculus. Does that change the telomere length? I don't know. And whether there are secondary mutations or loci that affect this, I don't know. It's something that's interesting to us, but I feel it's Peter's domain, and so they haven't explained it. And we haven't either. So I'm sorry, I don't have an answer.
Did you look at replication force in the TIM mutants?
Oh, good question. We're doing that at the moment. There's no obvious proliferation defects, so just gross looking at BRDU incorporation, cell cycle profiles, growth rates. They look relatively normal. I mean, I think it's difficult to compare the knock-in ES cells to this complementation system, but we've also now able to complement with the pitbox mutant, so we can do the direct comparison. But it's a very important point. But yeah, we're doing that at the moment.
Based on the telomere fragility phenotype and the fork collapse, have you looked at the interaction between the pitbox mutant and either Fanconi defective cells or Werner?
So making the cross. Yeah, I mean, it's quite a big experiment. But yeah, I mean, we're starting to think about crossing the pitbox knock-in mice to various other genotypes. I remember the list that I mentioned at the beginning, the synthetic lethals between artel and blooms, artel RECQ5, artel MUS81, artel FANCJ. We've made all of those mice now, and we've now made the double knockouts in a conditional context. We can see lethality in the worm. What we can't do is actually do a controlled experiment, which would allow us to dissect why they're dying, okay? And so that we're now in the position to do that. Now, what's really interesting with, for example, in the FANCJ case, and that's the one case where we have crossed in the pitbox mutant, you inactivate artel, and we had to do this where we have the pitbox mutant over the a floxed allele in the FANCJ null background. When we add Cree to inactivate the remaining null allele of artel, basically the cells just stop replication dead. There's no BRDU incorporation. We don't see any fork extension. It seems absolutely essential for replication. Really remarkable. And that's consistent with the cell lethality that we see in the worm, the dog one, artel double. Why that is, I don't know, but it really suggests that there's a profound redundancy between artel and FANCJ. And I think the interesting part there is that artel and FANCJ are the most related helicases to each other phylogenetically. And in Drosophila, there's no FANCJ, and artel is absolutely essential. So I think there's something really interesting going on between those two that we want to understand.
Yes, so your T circles that you saw increased were presumably in telomerase positive.
Correct, then.
Have
Yeah.
you looked in telomerase negative then?
We haven't, no, it's a good question. What we did show is that actually the SLX4 dependency on telomere circles is extended to all forms of alt cells. So for example, if we look in the VA13 alt cells and then downregulate SLX4, we completely suppress telomere circles in those alt cells. So we think this is a feature also shared with alt, and I think you're familiar with alt because you work with Roger. I mean, you probably think this is alt. We're inducing alt in mouse. It's
Telomere phenotype is the first
just look like.
yeah.
So we're actually thinking that downregulation of artel may be a prerequisite to alt, and Roger has evidence that ATRX is also doing this. And that's something that we're looking at, trying to re-express or produce moderate levels of artel in alt cells. Does that actually induce senescence? I mean, that's a possibility. And And I think other features of ALT are really interesting. So you start to see incorporation of subtelomeric sequences. Basically instead of TTA GGG, you see TCA GGG. So the telomere becomes heterogeneous and it starts binding to proteins it shouldn't. And actually, RTL has the right characteristics for that because it has a very high rate of illegitimate recombination, which would be required for the incorporation of these abnormal telomere sequences into the telomere.
And your glioma data is also
Exactly.
characteristic of all this.
Yeah, so glioma, as you're kind of pointing out, is actually a very high rate of ALT in glioma. I mean, it's a correlation at this point, but yeah, it's a very good point.
Then Shangmi.
Very nice work. And I have a question about RTL knockout phenotype. So RTL knockout is proposed to lead to excision of the telomere circle. I noticed some of the, on your spread, it seems some of the telomeres, they lost the signal on a single sister telomere.
Yeah.
So how do you, what's your hypothesis?
Are you Tisha in disguise? She always asks me that question. I don't know. I mean, I think we're seeing a snapshot. What I'm not showing you is, I always say telomere loss or telomere shortening, but actually, if you inactivate RTL and do quantitative telomere fish, you see basically the telomeres become very heterogeneous, not just short, not just lost, but you also see very long telomeres. So what we've actually shown is if we measure telomere sister chromatid exchange frequencies, they're dramatically increased when you get rid of RTL. So it's possible that when you excise the telomere by SLX4, then you're going to generate a double-strand break, which could be a recombination substrate to invade on other telomeres. And that would allow you basically to adopt basically lengthen the telomere and appropriately depending on the point of strand invasion into adjacent telomeres. Whether or not you could do that on a single, you're seeing just a single sister being lost, I don't quite know how that is. So yeah, it's not clear. And there doesn't seem to be a G or C strand bias.
Yeah, if you do the co-fish, you
Yes.
see a preference.
Right, and we don't see a preference.
The last question is Shangmi.
So, SLS4 is a PEP4 protein. It can interact different protein. So it seems it has multiple functions. But you delete the SLS4, you propose is the resolution lead to the telomere.
Oh, SLX4. Yes, yes.
But do you have any idea which kind of enzyme actually makes
Yes.
the...
Sorry, I mean, just in the interest of time, I didn't go into all of the details. SLX4 is obviously a scaffold, and that's the first thing we knock down because it gets rid of all of the nucleases. We then made the RTL MOS81 double because we thought it would be MOS81, and it wasn't. So that was in double knockout MEFs. You still see telomere loss and circle formation in the MOS81 RTL double. We also downregulated SLX1 and ERCC1. The other two, well, SLX1's the nuclease and ERCC1 the partner of XBF. And downregulation of SLX1 led to almost a phenotype, the SLX4 phenotype, so suppression of telomere circles and telomere loss. So we think that the SLX1, SLX4 complex is the most relevant, but ERCC1 also has a contribution as well. So we do see a reduction in the level of telomere circles and a reduction in telomere loss. And we've confirmed everything I've shown you by hairpins in double knockout mouse cells that we've obtained from John Rouse. So we've basically made the SLX4 RTL conditional and the SLX1 RTL conditional, and we see exactly the phenotypes that I've showed you. So it is the SLX1 nuclease that is the nuclease responsible here in conjunction with SLX4, with a minor contribution from ERCC1.
All right. Please join me in thanking